Good afternoon, everybody. I would like to welcome you to this interesting session on. You can hear me clearly?
Yes, we can.
Yeah, okay. So our session is on genomics of infection and immunology. We have three prominent speakers, and I would also like to welcome my colleague, Dr. Gulfran Khan, who will be a moderator with me on this session. My name is Moiz Bakhid. I'm a consultant neurologist and professor of molecular medicine and immunology. So after we go through our session, each one of our prominent speaker will have 30 minutes to give a presentation. Then we will go to the panel discussion for 15 minutes and closing remarks at the end. And I hope everyone has enjoyed this fantastic conference. So our first speaker is Professor Rifat Hamoudi, and his talk will be the use of bioinformatics and in silico analysis, followed by biological in vivo validation to decipher the molecular mechanisms of COVID-19 infection. Professor Rifat, he is a professor of molecular, cellular, and computational medicine at the University of Sharjah. He has 3 different MSc degrees from the University of London in Biochemistry, Molecular Biology, Electronic and Medical Engineering, and Computer Science, respectively. He has a PhD from University of Cambridge in Pathology and Immunology. It's very exciting areas of different specialties, and it's my pleasure to introduce you, Professor Rifat. I'm happy to see you again after we met last time on the Bahrain Genomic Conference. You are heartily welcome to make your presentation, and the floor is yours.
Thank you, Prof. So I'll try and share now. Sorry. Yeah, thank you, Prof. Moiz, for the kind words and the introduction. So my talk is today is going to be about maybe it's slightly different. It is to do with immunogenetics, but it's looking at maybe a different aspect of using bioinformatics and in silico analysis, followed by kind of in vivo and, you know, you could say in vitro validation to try and really understand some of the mechanisms involved in the SARS-CoV-2 infection that causes the COVID-19 disease. So before I go through the data and the results we found within the lab that I work in, I just wanted to kind of put a perspective on this, like COVID-19, what we call that problem formulation. So yeah, so COVID-19 came, well, it was declared as pandemic around February or March 2020. And since then, it's caused a huge burden, like we all know now, on the healthcare system and also the economy worldwide. But the problem with this, with the virus, the SARS-CoV-2, is that it has, you know, multiple spikes protein, and these can be used to morph, maybe in engineering terms, to morph the virus. So basically, it's very adaptable. So it seems to be much more adaptive than, you know, previous versions of SARS. And as we know, once there were many treatments, sort of maybe I'd say April, May onwards from 2020, and then the vaccines came around October, I think, during the summer of the 2020, they were doing some of the clinical trial. The issue, it seems that new mutation, the virus sort of, keeps escaping the treatments and also some of the vaccine by acquiring new mutations every 3 to 4, maybe 6 months. Yeah. So the mutations previously, the alpha, beta, I think delta, there used to be a single point mutations on some of the individual, spike proteins of the virus. But the more recent one, the Omicron, it showed that the virus can acquire multiple mutations on one of the, spike protein, and that, as we know now, that with the Omicron, that seems to make it more infectious, so it gives it this higher infection rate. But at the moment, we still don't know exactly whether this is it as severe as the previous one, and can it escape some of the vaccine? It's still a work in progress. So here, what we need really is to understand more about the mechanism of this COVID-19, but from the host perspective. So from how it what does it do in the host when in the human, like once the infection happens, what happens and how does it bring its devastating effect? So that's what I'm going to try and sort of do in this presentation. So in order to answer some of these questions, we used kind of like a bioinformatics approach with basically trying to analyze transcriptomic data to look at some of the molecular mechanisms involved in COVID-19. So for this, we took some publicly available data. These are from autopsies of COVID-19 infected patients that were mostly from Italy. Right at the beginning, if you remember, during the pandemic, I think many people died in Europe generally, but I think Italy had, a high percentage of mortality there. So they kind of deposited this data, and we took it and we applied, slightly innovative, different ways of analyzing the data. The data came from, different tissues. So we had lung, liver, and also there was renal and some heart as well from the same kind of cohort. But and these are the sort of, people are interested, you can go into this gene expression omnibus and download the raw data. So this is the raw data, but we did slightly different analysis to do the filtering and the differential gene expression, and that is then followed by some of the pathway analysis and mapping it to the disease gene. So what the data initially showed is that with COVID, there are different genes that seems to be more unique in some organs than others. So these like 23 that are unique to the liver, 55 to the lung, and so on. But then there are also common ones. So there are common genes that are across all the organs. So like an overview in the beginning, we saw that there are different the level of differential expression between basically the up and down regulated genes between the different tissue is more or less the same, you could say, but maybe in the kidney you have a little bit more on the upregulated gene, but it's not that huge. So the ratio is also the same roughly between the up and down regulated genes. When we map this to pathways, so what we found is obviously, and this is to no surprise anyway, that the virus, the top pathways are activated, are really to do with the immune system. So what we saw is that both the adaptive and also I think somewhere the innate immune system, and also the inflammatory response is kind of being activated as a result of the infection of the SARS-CoV-2 virus. So so it identified multiple different immune response pathways, but also many of these immune response, if you kind of look, like if you go for the genes, like for example, the regulation of cytokine-mediated signaling, many of them are to do with the chemokine and cytokine. But in addition, when you think of the inflammation activation, that's to do with the NF-kappa B pathway. So you have the NF-kappa B pathway and the response to pathogen, probably as a result of the NF-kappa B pathway shift. And then that shift might deregulate or dysregulate the process of the chemokine and cytokine expression as and when the cell needed. But in this case, it seems that the virus is somehow manipulating this mechanism to its own advantage. So then we kind of like went deeper into this, and we found that by looking at the individual genes, so the previous slide was the pathways. So here, what you can see is an interesting thing, that with the individual genes that were differentially, expressed between the tissue, you have a kind of a pattern. So you have some of the genes would be uniquely overexpressed in one tissue as opposed to the other. So, for example, the CXCL10 is upregulated in the lung in this case, but is downregulated in the other organ. But then the IL-1 beta, it's kind of downregulated in the lung, and it's usually upregulated in the kidney. But then it's sort of mild upregulation in the liver and the heart. So you get this kind of unique pattern across the tissue, and within the tissue here, you also have very different signatures. So so obviously the virus seems to be doing like kind of reprogramming things, but at a different level across tissue and within the tissue itself. So that was also interesting. And most of these that we found, they're also linked to the, the interleukins, the chemokine, interferon, and the TNF members, plus some of these markers are linked to the cytokine storm. So it was kind of an eye-opener, I guess, once you start to look at this kind of data. Okay, yeah. So then we sort of tried to do some mapping on the immune system itself, from the genes. And we found, this is just a very kind of qualitative way of looking at it, but we find that from the genes, we do get different response to, like, with different immunome data set. So in this case, we have the immune cells, the chemotaxis using the gene ontology, and here you find, that you're getting, for example, this one is high in the kidney. So if you're looking at the signature of the genes that are in response to specific immune cells, so macrophage, monocyte, T cell, and so on, you do find different, the immune cells will be activated differently between the various tissues. But generally, the T cells come up, I guess, in this set, which is coming from the immunome gene ontology, but then in other sets, you get slightly different. The reason why we use the different sets is just to cross-validate the data set that is formed by merging the different genes. So then we also looked at, we tried at least to investigate the inflammatory response. And what we found is that, it is also an interesting one, because it seems that a lot of the NF-kappa B subunit and also the inhibitors are kind of coming up in there. But as you know, with the NF-kappa B, it's really the degree of, because when you go into the subunits, you will not see a big change in these, because they shuttle between the cytoplasm and the nucleus. But you do see a slight fold change between them. And I think it's this, well, we think it's like, this kind of fold change that we get, although it's very tiny. It's shifting, the pathways is kind of shifting that transcriptional profile of the chemokine and cytokine slowly to bring on the devastating effect. So although here, for example, if you look at REL, you have strong expression in the lung and also probably the kidney, but less in the liver and the heart, but they're all like overexpressed. But these are the subunits, so, and when you compare that with the chemokine and cytokine, which are most of them are NF-kappa inducible, you start to put a little bit of the picture of what's going on in there. Okay, so then obviously this is from the in silico stuff. So once we kind of got this pathways and genes, now we wanted to see if we can get some biomarkers to kind of predict maybe the severity of COVID based on this type of analysis. So these two tables, they show that there are different biomarkers that are uniquely linked to the different organs in there. So once we identified those biomarkers, then we obtained some blood and saliva samples from COVID-19 infected patients, and here you can see the results. So what we did is then qRT-PCR on these, and here you can see that some of them they do predict the severity of the COVID-19 by looking at some of the markers across the different subtypes of the COVID-19 infected patients, and also with the different organs as well. So okay, so in conclusion for this one, what we found is that basically it's quite a complex thing. So it starts from the lung, the COVID-19, then it reprograms, you kind of have this transcriptional shift, probably by dysregulating the NF capillary pathway. And from that, it seems to bring on what is probably some sort of fluctuation in like recurrent cytokine waves. So you generate the cytokine wave, and then the body tries to overcome it, but then it keeps doing that. So it's almost like an amplification of the signaling of the immune response that keeps happening. And then at the end, if the body can't recognize this, then it transmits this effect onto other organs. And that's why some of the patients they have, it's not I mean the multi-organ failure, there are some patients that did report that. But as you know, in the beginning, there were a lot of patients who had, keep dialysis. They would go on dialysis. Some would have heart attack, and others they had some liver failure. And also the long COVID. But we didn't, because we don't have really neurological, sample like from the brain. I don't know. We didn't see that in this data set, but it probably, this shift in the immune response through the alteration of these chemokine and cytokine, but uniquely to different organs, it probably will also affect the nervous system as well. So that work is published in the Frontiers of Immunology, but then as well as that, we also kind of were interested in looking at the effect of the same phenomena, but on the liver. So what happens really at the liver? So again, we compared the liver with the lung, in this case, because it starts from the lung. And we found that there are different pathways that are being, activated in these data set. So from the pathways, this is just the same as the other, but we're looking at the pathways, like the expression of them between the healthy and the COVID from this data set. So we identified some interesting, pathways that are linked to liver function. Then we again got some samples, both from the collaboration within the UAE, and also we had some collaborators from Germany. And when we did the RNA-seq, or this is the some of these are RNA-seq and some of them are qPCR. So we found that yes, some of the genes, they do show the trend that in the severe with liver dysfunction, you do get higher expression of the gene as compared to the healthy and or the severe. So in the liver, the story is that it seems that these genes are downregulated, and those genes are linked to mitochondrial function and metabolic pathways, and also hepatic detoxification and drug clearance. So what it means is that when they're regulated, then you get less of the detoxification, which is what the liver is the main liver function is that. And plus this ion transport, and homeostasis. But really, it's that hepatic detoxification that is the big issue here. And then you also get upregulation of those genes which are linked to tissue remodeling, liver enzyme levels of circulation, and hepatotoxicity and liver dysfunction. So what it seems is almost like you're getting, like kind of cirrhosis, but as a result of the infection with COVID. So this study was published in World Journal of Gastroenterology. So from these multiple genes, what we wanted to do finally is to try and see if we can validate some of the finding using, because you saw that we tested things on patients and it came out true. But we wanted to see, like, for example, can we understand why this virus is manipulating these molecules to do with the immune system? So one of the markers we got is this one, is a C1q-TNF. And this is like, it stands for complement C1q, which is part of the innate immune system. But this molecule is thought to also to be acting as an adipokine, and it modulates various metabolic processes, and therefore, because of that, it links to cardiovascular, probably pulmonary and renal function. So what this was our strategy, that what we wanted to do is to try and do some of this functional analysis. So we got the vector, so we got the gene code in an expression vector, and then what we do is we transfect that into lung fibroblast in the one that I'm going to show you. But what we did is we used diseased human lung fibroblast and also normal human lung fibroblast. So this is just checking that we are getting the C1q to be overexpressed before we do the next generation sequencing. And also this is doing it like checking that gene on the patient, and you can see that it is upregulated in the severe patients as compared to mild in the control. So we run the NGS, and this is just the quality control of it. But when we did the analysis, so here this is looking at overexpression of the C1q in the normal lung, and also the same gene but in the diseased. The diseased lung is sort of like more like asthma, this DHLF. So these are, you can get them from Lonza, it's commercial one. So then we looked at the genes, and the top ones differentially expressed, I guess, are these ones. This is just checking it with the qPCR. And you can see that there are different level of expression, both to do with the time, based on the time point of the transfection, and also across the different types of the cells. So then finally, we again looked at the pathways for this. And when we did some of this analysis, we did find that in the normal, when you overexpress it, it does bring you the signature of SARS. So it's not the COVID-19, but it's generally the virus, this signature. And also it brings the response to virus. So it seems to be doing things that are linked to response to virus, and also the signature of the gene. Plus in the asthmatic cell or the disease cell, we also get the viral signature, which is the influenza A, and also we get seem to be getting this interferon alpha beta signaling as well to come up. So there are different types of response, coming out, but it is interesting that once you transfect it, purely just transfecting it, you're getting the signature of the SARS-CoV-2. So in conclusion, then the bioinformatics algorithm, the one or the methodology that we used here, it was able to identify, differentially expressed genes that are linked to immune response in addition to other cellular pathways. So, like you saw, there's metabolic, but majority of them are linked to, the immune response. Now, and the biomarkers that are identified using in silico analysis, they're also shown to be differentially expressed between the tissue and across the tissue. And this really explains maybe some of the complexity of this virus, because it seems to be, heterogeneous. It can adapt, and, it's sort of looking at the overall picture, so it gets shed some light on the heterogeneity and the complexity of COVID-19. Yeah, and whatever. So again, if you're doing bioinformatics, you have to see if it carries on to translational. So in this case, we have shown that majority of the markers that we picked up when we tested them on blood and or saliva from patients with different COVID-19 severity, they were validated. So they are linked to severity. So they're either, upregulated with severe cases or extremely downregulated in some cases. Then for the validation, so when we looked at the in vitro validation with cells, again, it seems that, when we transfect, in this case, we just looked at the C1q, but we're in the process of looking at other markers as well. But when you just, transfect this gene, you do get the signature for SARS-CoV-2. So I guess here is just to show that there is, because COVID is really quite a complex disease now. Now, for all of us know, because we think that, oh, we are winning, and then suddenly a mutation come up, like the Omicron case. So I guess if one adopts a multidisciplinary approach, we might be able to, try to have some more insights into the mechanism of the virus, and then it will give us better idea of whether, like say if we get a new mutation in the next 3-4 months after Omicron, if we adapt this strategy, it will tell us probably straight away whether the new mutation or the new variant is that going to be, causing like hospitalization and things like that, or is it going to be just a mild, infection? So the idea here is like apply the multidisciplinary, but looking at host transcriptomics rather than the viral or the host genomics alone. A lot of the studies, they use more the viral genome, which is obviously important, but I think we should couple this with the host transcriptome to really understand the virus, because with the immune system, it's always like give and take. It's sort of more like trying to get two ecosystems to either work together, and if they don't, what is the problem? So that's the idea of doing this type of work. So then the idea, the final point is just to try and see whether some of these biomarkers, can we use them to see whether the new mutations are they very severe or not. So I'm sure we'll probably get some more mutations to do with the SARS-CoV-2. So with this, I'd just like to acknowledge maybe Dr. Habib Al-Safar, who from Khalifa University, who provided very good sample sets to do the validation of some of the findings that we got from the bioinformatics. And obviously University of Sharjah for support of the work, and Sheikh Dr. Sultan Qasmi for his endless support of all these kind of research that we do at the university. Okay, so thank you very much. Thank you.